Hello everyone, welcome to the video lecture series on Advanced Separation Process. Myself, Jamin Pandya, Assistant Professor in Chemical Engineering Department of LJ Institute of Engineering and Technology. So let's continue with the chapter number 2, which is a membrane separation process. So in this lecture, we will discuss about various techniques for the characterization of membrane. So in previous lecture talk about the TEM which means transmission electron microscopy. Apart of TEM there are two more techniques for microscopic method such as HCM and AFM. HCM which means scanning electron microscopy and AFM which means atomic force microscopy. So let us understand what is the difference between TEM, SEM and AFM. So let's first revise the TEM technique. TEM technique operate under high vacuum. So all setup or can say that entire setup of TEM is operate under high vacuum. We discussed why we are going to use high vacuum in previous session. So let's just revise it. So this is one electron gun which provide electron to the specimen. This specimen which is our sample such as a membrane or other polymer material. So this electron gun spread the electron from condenser 1 and second condenser lens. So this electron pass from the first condenser lens, then go to the condenser apparatus and then pass to the second condenser lens. Then is passed from the specimen which is our sample such as a membrane, and this electron pass through this specimen or can say that sample membrane and generate electron beam which spread on the surface or can say that fluorescent screen or camera and generate the image. A electron that pass from this sample is known as a scattered electron, and a electron that reflected from the sample is known as a unscattered electron. Unscattered electron make image black, where scattered electron make clear image. So here we are going to provide high vacuum system, because if we are going to provide a normal pressure, so electron there is a chance for electron that we collide with each other and we did not get the clear image. So this is the purpose of providing high vacuum system in this entire setup. So this is one example of image of from getting from the TEM or can say that transmission electron microscopy. So this is one example of TEM for the polyamide membrane, and this is the original setup for the TEM. So this is all about TEM. Now let's discuss about the SEM, which known as a scanning electron microscopy. In scanning electron microscopy, electron sources are provided from which electrons are scattered or can say that bombard or strike to the condenser lens. There is two condenser lens. There is a this is one primary condenser lens and this is secondary condenser lens. So this electron beam is passed from this anode to the primary condenser lens and secondary condenser lens. Then it passed from the objective lens and scatter on the surface of the sample or considered specimen. After striking the electron on this surface is detected by the X-ray detector and generate the image in the PC or computer. So this generated image is sensed by the X-ray detector and the secondary element detector, which amplify the image and generate into the computer. The main purpose of this condenser lens or can say that various lens is that it magnifies the image. So basically there is a electron source which provide the electron which pass from the anode and condenser lens system, then it pass from the objective lens, then it pass to the surface of the sample and rejected. So we will get scattered electron which generate the image and unscattered electron which generate the black image. So this is one SEM image of the polyamide membrane, where here we can see the clearly a pore of the membrane, and this is a cross section of the membrane where we can clearly see here there is a cylindrical pores throughout the membrane. So what is basic difference between the SCM and TEM? In SCM, we are just going to scan the surface of the object, or can say that sample. So in FCM, we are just going to scan the surface of the object or sample, where in TEM, we are going to scan entire sample. So in TEM, the electron is passed from the surface of the sample. So let's consider this is one sample, or can say that specimen, or can say that our membrane. So if we want to characterize this membrane, electron pass through the this membrane in TEM, where in SCM just electron is scattered on the surface and rejected. So in SCM we are just scanning surface of the membrane, where in TEM electron pass through the membrane. So this is a basic difference of the TEM and SCM. So this is example of real SCM system, and this is the inside area of the scanning electron microscope, where the electron gun provided which pass through the anode and various lenses to magnify the image and pass through the sample. From this sample it detects from the X-ray detector and the secondary electron detector, and it's sent to the computer. So this is all about scanning electron microscopy. Now let's discuss about the atomic force microscopy. The atomic force microscopy has demonstrated a wide range of application in investigation of physical characteristic of surface. Again, AFM is a alternative of SCM that scan the surface of the membrane or sample. So this is one example of the atomic force microscopy. In atomic force microscopy, the light microscope are provided, laser diode are provided, from which laser is reflected and passed to the cantilever. This cantilever is near to the sample surface, so it senses the surface by touching it or by the tapping it. And there is another method for the cantilever which senses the surface without touching by the mode of the frequency. So using frequency manner we can or amplitude manner we can also get we can also get image from the AFM. So there is two methods for sensing the surface in AFM. One with contact with surface, and or can say that by touching of surface, and second method is without touching of the surface or without contacting of the surface. In without contacting we are going to use the frequency or amplitude, where in a touching method we are this cantilever oscillated and touch the surface of the sample and sense the various bond and various roughness. So mostly AFM are used to demonstrate the roughness or to scan the surface of our sample. But AFM is also used for the generate image of various liquid, organic solvent, solid, where SCM is restricted only for the solid material, where atomic force microscopy which also used for the understand morphology and geometry of liquid surface. So what is basic difference between SCM and AFM? We know that the what is difference between SCM and TEM. In SCM we are just getting surface morphology, where in TEM electron pass from the sample and we are getting whole information about the surface and other component, where in SCM we are just getting information about the surface. In AFM we are also getting information about the surface, but we are getting 3D diagram just like this compared to the SCM. So this is one example of SCM image, and this is one example of the AFM image. In atomic force microscopy we are also get the information about the various force such as a van der Waals force between two molecule or atoms. So we can also get this information from the atomic force microscopy. Using frequency or amplitude, cantilever sense the force between two atoms and reflect in a image form. Then it's passed to the PSPD detector and in the computer. So we will get the image like this of the membrane surface, where this is a force of the membrane surface, and this is real setup of the AFM. So this is all about atomic force microscopy. Now let's talk about the SCM versus TEM. SCM used to produce excellent image of the surface of cells and small organism and membrane, excellent for studying surface morphology of the various membrane and cells or any suitable material under study, where TEM, or can say that transition electron microscopy, used to study ultrastructure of the cell and its component. It can see the object as small as a protein molecule or even at nano level, provide detail about the internal composition of cell or any suitable material under study. So this is one example of the HCM and TEM. So this is the image or using the HCM, where this image is for the TEM. So this image is for the TEM, or can say that transmission electron microscopy. So from the SEM we are just getting information about the surface, but using TEM we can get detailed information about this molecule. If we want to know about this molecule, this is detailed image of this molecule using TEM, where can easily understand this is a wall of the cell and what is the inside structure of this cell. So TEM gets ultra magnifying image compared to the scanning electron microscopy, but it's consume higher energy compared to the scanning electron microscopy. So if we want to just understand surface morphology, we are going to go through the SCM, but if we want to understand the detailed structure of the membrane or what is the various component and what happen in the membrane, inside the membrane, then we have to go for the TEM, or can say that transmission electron microscopy. In SCM, electron scan over the surface of sample, where in TEM electron beam pass through the sample. So in SCM, electron beam cover from the surface of sample, where in TEM it pass from the sample. So this is one sample where electron pass from the sample, and here just scanning over the sample. In SEM, based on scatter electron or produce image by detecting secondary electron which are emitted from the surface due to excitation by the primary electron, where in TEM based on transmission electron or produce the image by detecting primary electron transmission from the sample, where in SEM comparatively low resolution than TEM. Resolution in SEM is near about 2 nanometer or 0.2 nanometer for the spatial SCM, where in TEM high resolution we will get high resolution near about the 10 nanometer to the 0.5 nanometer, and for the SCM we are getting depth of field or depth of field which is a high, where in a TEM depth of field is a moderate. Magnifying power of the SCM is a 1 lakh x, where magnifying power of TEM which is a 50 lakhs. Specimen contract by the electron absorption in a SCM, while in TEM electron scattle are used. In SCM produce 3-dimensional black and white image, where in TEM producing 2-dimensional black and white image. In SCM preparation technique is easy, where compared to the TEM. In TEM we need skill, very thin sample is required. In TEM, as we discussed, there is electron pass from the membrane or sample. So we require very thin layer of the sample. So electron can pass over this membrane or can say that or that sample. So we require special type of coating on sample and very thin layer generation of that sample. In SCM preparation thickness is a variable or it might be changed, where in TEM thickness of sample is very thin. In SCM specimen mounting in aluminum stub, where in TEM thin film on copper grid are provided. In SCM field of view is very large, where in TEM field of view is very limited. So this is a basic difference between scanning electron microscopy and transition electron microscopy. So in this lecture discuss about the various methods, such as the microscopic method for the characterization of membranes such as the SCM, TEM and AFM, which use for the generate image of surface and cross section, or understand the bonding between the membrane, or generate image of cross section of membrane or foulant, or it's also used for the generate image of roughness surface morphology. Then discuss about the transmission electron microscopy, SCM, which means scanning electron microscopy. Then talk about the atomic force microscopy. In atomic force microscopy, cantilever are used to sense the surface, and there is two techniques for the sense this surface. One which is a without contacting, and second which is a with contacting. In without contacting, amplitude or frequency are used, where in contacting there is a cantilever are used to sense the surface layer. So this is the image of AFM, this is image by the SCM, and this is image by the AFM. In AFM we can get the detailed image, where in SCM we will get black and white image like this, and in AFM we will get the 3D image. So this is all about today's lecture. In next lecture we are going to discuss about the various other techniques for characterization of membrane, such as bubble point mercury and various other methods to understand information on membrane pore structure. So this is all about today's lecture. I am completing my lecture here. Thank you.